Hey everyone, and welcome to This Explainer. If you've ever wondered how modern surgery achieves that absolute, perfectly motionless precision, well, the secret actually lies in mastering a really fascinating pharmacological tightrope. We're talking about muscle relaxants. So today, guided by the incredibly detailed research of Professor Sanjeev Singh, we're going to dive deep into the actual mechanics of these incredibly powerful drugs. But first, let's look at the fundamental problem here. Deep anesthesia all by itself? It just isn't enough. To safely place a breathing tube, facilitate mechanical ventilation, and, crucially, secure those perfectly still operating conditions that surgeons absolutely need, medical professionals have to rely on precise neuromuscular blocking agents. So here's our roadmap for today. We'll start with the physiology of the NMJ, move into depolarizing and non-depolarizing agents, then look at reversal, assessing recovery, and finally wrap up with our key takeaways. All right, part one, the physiology of the NMJ, basically understanding what normal looks like. So picture this. Inside the nerve terminal, you've got these little vesicles, and they are packed with a neurotransmitter called acetylcholine. When an action potential hits, a rush of calcium triggers these vesicles to essentially dump their payload right across the gap. It's this beautiful lock-and-key mechanism. The acetylcholine binds to specific receptor sites on the muscle membrane, pops open the voltage-gated sodium channels, and boom, you get a muscle contraction. Understanding this normal physiology is absolutely vital because literally every single drug we're about to talk about hacks this exact biological hardware. Which brings us to part two, depolarizing agents, specifically succinylmethanonium. The absolute star of this category is succinylmethanonium. You might also know it as succinylcholine. Structurally, it's actually just two acetylcholine molecules glued together. This makes it an agonist, so it forcefully binds to the receptors just like normal acetylcholine would, causing a massive depolarization. Clinically, if you're watching a patient, this actually looks like a brief, visible rippling of the muscles right beneath the skin. We call those fasciculations. But here's the kicker. Succinylmethanonium absolutely refuses to leave the receptor, so the muscle simply exhausts itself. It basically gives up and goes into complete flaccid paralysis in just about 30 to 60 seconds, super fast. But you know that incredible speed? It comes at a really high cost. Because the receptors are forced open and held there, potassium literally pours out of the muscle cells and straight into the bloodstream. Now imagine a patient with severe burns, crush injuries, or spinal cord damage. Their bodies try to adapt by growing millions of extra immature receptors all over the muscle surface. If you give succinylmethanonium to these patients, you get a massive, lethal spike in serum potassium, which can trigger instant cardiac arrhythmias. Honestly, it's terrifying, which is exactly why this drug carries such strict contraindications alongside other serious risks like malignant hyperthermia and profound bradycardia. Let's pivot to part three, non-depolarizing agents, our competitive antagonists. Okay, so unlike succinylmethanonium, which remember forcefully activates and holds the receptor open, non-depolarizing agents take a completely different approach. They are competitive antagonists. I like to think of them as giant bouncers standing right in front of the receptor's door. They don't activate the muscle at all. Nope, they just sit there on the pre- and post-junctional receptors and physically block the natural acetylcholine from ever getting in. Resolving this kind of blockade relies purely on waiting for the drug to eventually diffuse away from the junction into the plasma, where the body can clear it out. Within this class, we generally look at two main chemical groups. First, the aminosteroids. This includes pancuronium, which lasts a really long time, like 40 to 50 minutes, and is cleared mostly by the kidneys. Then there's vacuronium, which is shorter acting and relies heavily on the liver. But then we have the benzyl isoquinolinium group, which includes atracurium. And atracurium is a real game changer. Instead of relying on the liver or kidney enzymes to clear it out, it uses something called Hofmann elimination. It literally undergoes spontaneous chemical degradation in the blood plasma, based purely on the patient's normal body temperature and pH. It's like a pharmacological silver bullet, making it exceptionally safe for patients who are suffering from multi-organ failure. But of course, anesthesiology is a delicate balancing act, right? Several environmental states and even concurrent medications can drastically prolong this blockade. For instance, if the patient is hypothermic or acidotic, the block is going to last longer. Surprisingly, even common aminoglycoside antibiotics, like gentamicin, can inhibit acetylcholine release, which deepens the paralysis. And naturally, patients with myasthenia gravis, an autoimmune condition where their antibodies actually destroy their own acetylcholine receptors, well, they're going to be profoundly sensitive to these competitive bouncers, often requiring just a tiny fraction of the normal dose.
Okay, part 4, reversal and anticholinergics, waking the patient up.
So, once the surgery is totally complete, we can't just leave those non-depolarizing bouncers hanging around occupying the receptors. We have to initiate a pharmacological tug-of-war. We administer an anticholinesterase drug, typically neostigmine. Now, by inhibiting the body's natural cleanup enzyme, neostigmine allows this massive flood of native acetylcholine to build up in the junction. This huge concentration gradient essentially overwhelms and physically outcompetes our blocking drug. But here's the catch. This systemic flood of acetylcholine also hits muscarinic receptors everywhere else in the body. That can cause severe, even fatal drops in heart rate and massive salivation. Because of that, we absolutely must instantly pair the neostigmine with an anticholinergic.
When it comes to choosing that vital, heart-protecting anticholinergic, the main options are atropine and glycopyrrolate. Atropine works incredibly fast, but its major flaw? It crosses the blood-brain barrier. In large doses, that central nervous system penetration can cause patients to wake up with postoperative excitement, hallucinations, or severe drowsiness. Glycopyrrolate, on the flip side, is a larger, synthetic molecule. Because it physically cannot cross the blood-brain barrier, the patient avoids those central nervous system side effects completely. They experience far less tachycardia and generally wake up much, much smoother. Moving on to part 5, assessing neuromuscular recovery. How do we prove safe function? Before we even think about safely removing that breathing tube, we have to objectively prove that muscle function is fully restored. Now, while nerve stimulators do provide great electrical data, clinical milestones are really what it's all about. We're looking for a coordinated pattern of breathing between the diaphragm and the intercostal muscles. We test for a strong hand grip or the ability to bite down firmly. But the ultimate clinical stress test, asking the patient to lift their head completely off the bed, totally unassisted, for 5 full seconds. If they can nail that, it's a great sign their airway reflexes are generally intact and they're safe to extubate.
Finally, part 6 are key takeaway points. Let's summarize the whole shebang. Mastering these distinct pharmacological profiles is literally the cornerstone of safe anesthesia. On one hand, we have the depolarizing agonists like suxamethonium. Unmatched for sheer speed during emergency intubations for sure, but definitely burdened with lethal potassium risks. Then we have the non-depolarizing competitive antagonists. These range from organ-dependent clearers like vecuronium to the incredibly unique auto-degrading atracurium. And finally, you have that precise reversal choreography, flooding the neuromuscular junction with acetylcholine using neostigmine, while meticulously protecting the heart and the brain with targeted anticholinergics like glycopyrrolate. It's quite the balancing act. So I want to leave you with this final, kind of provocative thought. While we can skillfully navigate these hyperkalemia risks, unpredictable drug durations, and the whole complex tug-of-war of reversal agents today, the true future of patient safety lies in innovation. So what does the perfect, side effect-free muscle relaxant of tomorrow look like? Will it be instantly reversible without even needing those heart-altering anticholinergics? As you keep exploring pharmacology, keep an eye on the horizon, because the quest for the perfect neuromuscular blockade, it is far from over. Thank you so much for joining me for this explainer, and keep asking those great questions.